millions of patients are waiting for tissue transplants or effective cancer therapies. Tissue and tumour engineering are strategies addressing these problems by growing cells in the lab. The idea is to mimic the complex natural environment by three-dimensional cell cultures that could also serve as more reliable models for drug testing than the current monolayer cell cultures and animal experiments. Yet, these 3D cultures require effective supply of oxygen, nutrients, and biochemical signals, which is achieved in vivo by a highly branched network of capillaries. In a 3D culture, this can be simulated by providing a direct fluid flow through the culture. The single-use perfusion bioreactor 3D Perfuse is designed for this purpose and particularly suited for the use by scientists without technical expertise. The system is provided sterile and assembled in a pouch, which is easy to open in a laminar hood. The bioreactor cartridge consists of silicone tubing coming in 3 diameter sizes, 8 to 12 millimeters, and could be cut to the length appropriate for the particular sample. The bioreactor chamber is open at the inlet side, so that the sample with cells can be placed inside by forceps. The system contains 2 3-way stopcocks for accommodating 2 syringes filled with medium. After closing the bioreactor chamber, it is filled with medium by the inlet syringe, which is then used to fill also the tubing coil and reservoir. Finally, the tubing between the chamber outlet and the medium reservoir is filled with medium by a syringe placed in the outlet stopcock. Medium volume in the system is usually about 10 to 15 milliliters. The system is secured on a stand, which is then placed in a humidified CO2 incubator. The stopcocks are open so to allow medium circulation through the system provided by a multi-channel peristaltic pump. Several systems can be run in parallel under the same conditions. Medium can be exchanged as required by using a filter at the reservoir so that the culture can be maintained for longer periods of time, weeks, and even months. So, if you want to establish a 3D cell or tissue culture, or to test novel biomaterials in a physiological-like environment, the bioreactor 3D Perfuse is simple to assemble and apply. We are a multidisciplinary team experienced in setting up and conducting different kinds of 3D cell and tissue cultures, enthusiastic to answer all your questions.